Cell division. Cell division, or reproduction, is when a parent cell replicates its genome and separates into two or more daughter cells. Prokaryotes achieve this via binary fission, asexual reproduction, which was covered in an earlier unit. Eukaryotes undergo cell division using two different nuclear division processes. In mitotic cell cycles, a cell produces an identical copy of itself with the same number and patterns of genes and chromosomes. This occurs in somatic or non-sex cells. Meiotic cell cycles create gametes, sex cells like sperm and eggs, with half the number of chromosomes of the original cell. The gametes are unique, non-identical cells and are used for sexual reproduction. Cell division. Organisms undergo cell division to repair damage, as in scars or dead cells, regenerate lost parts, as in the lizard who loses its tail, grow in size, reproduce asexually, and reproduce sexually. Eukaryotic cell cycle. The eukaryotic cell cycle has two major divisions, interphase and the mitotic phase. During interphase, the cell metabolic activity is very high. It is busy growing and copying its DNA and organelles so it can divide. The mitotic phase includes two events, the division of the cell's nucleus, which is mitosis, and the division of the cell's cytoplasm, which is cytokinesis. Mitosis involves a series of steps, or subphases, as summarized in the chart on the right. Interphase. Most cells spend more than 90% of their lifetime, their cell cycle, in interphase. Interphase has three distinct subphases, which include gap 1, or G1, synthesis, or S phase, and gap 2, or G2. Gap 1, or G1 phase. In this phase, the cell increases in size. The cell increases its supply of proteins, particularly those used in the duplication process, and duplication of organelles occurs. Synthesis, or S phase. In this phase of the cell cycle, DNA replication occurs. At the end of this subphase, the number of chromosomes in the cell has doubled. Chromosomes cannot be seen individually because they have not yet condensed. Gap 2, or G2 phase. The cell completes its growth in preparation for division. It checks its DNA for any damage that might have occurred during replication, which is very accurate, by the way. It could correct one base pair out of billions of base pairs of DNA, and it ensures all proteins needed for cell division are present. Mitotic phase. After a cell completes its preparation for division, it enters the mitotic phase. There are two subphases to this phase: mitosis, the division of the nucleus, and cytokinesis, the division of the cytoplasm. Subphases of mitosis. Mitosis is further broken down into 5 subphases. These 5 subphases here, prophase, prometaphase, metaphase, anaphase, and telophase, are 5 subphases of mitosis that are all involved with cell division. Prophase. In this subphase of mitosis, the nucleolus disappears. Chromatin condenses into chromosomes, which appear as two sister chromatids joined at their centromeres. This is where we begin to see these familiar X-shaped chromosomes appear. Centrosomes start to travel to the opposite ends or poles of the cell, and arrays of microtubules called spindles start to form from two centrosomes. Prometaphase. The nuclear membrane disappears. The spindle is nearly completed and ready to provide a scaffold for chromosomes to travel. Chromosomes attach to the spindle at their kinetochores, a protein structure at the centromere region of the sister chromatids. Centrosomes versus kinetochores. This is an image of a human cell during division showing spindles from the centrosome in green, chromosomes in blue, and their kinetochores in pink. Notice how this image looks strikingly similar to our diagram. Centromeres and Kinetochores. The centromere region is the central part of a chromosome where the two sister chromatids attach, and these sister chromatids are identical copies of one another, which were produced during the S phase. The kinetochore is a protein structure in the centromere region where the kinetochore spindle fibers attach. Metaphase. In metaphase, the spindle is completely formed. The chromosomes align on the metaphase plate, which is the equator of the cell. Anaphase. In anaphase, the sister chromatids separate from each other at the centromere and are pulled to the two poles by the spindle fibers. Telophase. In telophase, cell elongation continues. The nuclear envelope reappears around the chromosomes, and the nucleoli reappear. Cytokinesis. Following telophase, the cytoplasm divides in a process called cytokinesis. Cytokinesis differs for plant and animal cells. Click here to watch a video showing the steps of mitosis. Cytokinesis. In animal cells, a ring of microfilaments forms a contractile ring around the outside of the cell. The ring forms a cleavage furrow which splits the cytoplasm in two. Because animal cells have a flexible cell membrane, we could pinch those membranes together and pinch off the cytoplasm, forming two new cells after the process of cytokinesis. Cytokinesis. In plant cells, vesicles containing cell wall material collect in the center of the cell and then fuse together. The cell plate forms from the inside out and turns into a wall between the two new cells. The membranes surrounding the vesicles fuse to form new parts of the plasma membrane. Remember, plant cells have a rigid cell wall that's not flexible like a cell membrane. Therefore, plant cells can't pinch in like the way an animal cell does. Instead, the cell plate must come and form a new wall before the two new cells can separate. Phases of the cell cycle summary. The interphase, the part of the cell cycle where the cell is not dividing and performing its functions, includes the GAP1 or G1, synthesis or S phase, and GAP2 or G2. The mitotic phase or M phase is where the cell divides, which includes mitosis, and the subphases of that include prophase, prometaphase, metaphase, anaphase, and telophase. And then there's cytokinesis, which is involved in division of the cytoplasm, as opposed to mitosis, which is division of the nucleus.